Advances in molecular biomaterials for tissue engineering. Advances in molecular biomaterials for tissue engineering. Tissue engineering is a rapidly evolving field that involves the repair and replacement of damaged or diseased tissues and organs. The goal of tissue engineering is to create functional biological constructs that mimic the structure and function of native tissues. One of the critical components of tissue engineering is the biomaterial used to create the scaffold for cell attachment, growth, and differentiation. Advances in molecular biomaterials have paved the way for the development of more effective tissue engineering strategies. Molecular biomaterials are synthetic or naturally occurring materials designed to interact with biomolecules and cells in a precise and controlled manner. These materials offer several advantages over are traditional biomaterials and have the potential to revolutionize tissue engineering. One of the most exciting areas of molecular biomaterials is the development of biomimetic materials that mimic the extracellular matrix, ECM, of native tissues. The ECM is a complex network of protein, NS, glycoproteins, and proteoglycans that provide structural support and biochemical cues to cells. Biomimetic materials can replicate the specific architecture and biochemical composition of the ECM, enabling cells to differentiate and form functional tissues. Researchers have developed several strategies to create biomimetic materials. One approach is to use self-assembling peptides that form nanofibers similar to those found in the natural ECM. These fibers provide an excellent substrate for cell attachment, migration, and differentiation. Another approach is to use decellularized ECM from animal or human tissues. Decellularized ECM provides an natural microenvironment for cells and contains the necessary biochemical cues for tissue regeneration. Another critical area of molecular biomaterials is the development of smart biomaterials. Smart biomaterials are materials that respond to external stimuli, such as temperature, light, pH, or electrical stimulus. These materials have several benefits for tissue engineering, including the ability to control cell attachment and proliferation, promote tissue regeneration, and deliver therapeutic agents. One example of a smart biomaterial is thermo-responsive hydrogels. Thermo-responsive hydrogels are hydrophilic polymers that form a gel at low temperatures and dissolve at higher temperatures. This property allows for the incorporation of cells at low temperatures and the release of cells at physiological temperatures. Additionally, these hydrogels can be used to deliver therapeutic agents by incorporating them into the hydrogel matrix. Another example of a smart biomaterial is electroactive biomaterials. These materials can be stimulated by electrical impulses to promote cell attachment and proliferation. Additionally, electroactive biomaterials can be used to control the release of therapeutic agents and induce cell differentiation. Molecular biomaterials can also be used to create microenvironments that mimic the native tissue microenvironment. Tissue microenvironments play a critical role in cell behavior and tissue regeneration. By recreating the microenvironment, researchers can promote tissue regeneration and repair damaged tissues. One example of a microenvironment created using molecular biomaterials is the use of gradient biomaterials. Gradient biomaterials contain a spatial variation in biochemical cues, such as growth factor, RS or adhesion molecules. These gradients can be used to direct cell behavior and promote tissue regeneration. Another example is the use of microfluidic devices to create microenvironments. Microfluidic devices can create precise and controlled microenvironments that mimic the native tissue microenvironment. These devices can be used to culture cells and tissues in vitro, allowing for the development of more effective tissue engineering strategies. Molecular biomaterials can also be used to create hybrid materials that combine the benefits of different material classes. Hybrid materials can improve the mechanical strength and biological properties of the scaffold and can be tailored to specific tissue types. One example of a hybrid material is the combination of natural and synthetic polymers. Natural polymers, such as collagen or fibrin, can provide biochemical cues and a biocompatible substrate for cell growth. Synthetic polymers, such as polylactic co-glycolic acid, PLGA, or polyethylene glycol, PEG, can provide mechanical strength and control degradation rates. Another example is the use of inorganic nanoparticles to enhance the biological properties of the scaffold. Inorganic nanoparticles can improve the mechanical strength of the scaffold, promote cell proliferation, and control the release of therapeutic agents. Furthermore, the development of molecular biomaterials has also led to the creation of more personalized healthcare solutions. Personalized medicine aims to tailor medical treatment to an individual's specific needs and traits. Molecular biomaterials can contribute to this field by providing tissues that closely match the patient's native tissues. This can reduce the risk of rejection and improve the overall success of the treatment. However, several challenges must be overcome to fully realize the potential of molecular biomaterials. One significant challenge is the scalability of the manufacturing process. Many of the current molecular biomaterials are produced in small batches, which can limit their commercial viability. Additionally, regulatory approval is another hurdle to overcome, as molecular biomaterials are often considered unique and require extensive testing before approval. Another challenge is ensuring the long-term stability and safety of molecular biomaterials. Many of the current molecular biomaterials have not been extensively studied for their long-term effects, and potential risks may not be fully understood. The safety profile of molecular biomaterials needs to be validated through rigorous testing before widespread clinical use. Despite these challenges, the potential of molecular biomaterials for tissue engineering is undeniable. The ability to create precise and tailored biomaterials that interact with cells and tissues in a controlled manner represents a significant breakthrough in the field of tissue engineering. The continued development of molecular biomaterials will undoubtedly contribute to the creation of more effective and personalized healthcare solutions in the future. Moreover, the use of molecular biomaterials can also contribute to reducing the reliance on organ transplantation. Currently, many patients with organ failure have no other option than waiting for a transplant from a compatible donor, which can take a long time and poses several risks. With the development of tissue engineering using molecular biomaterials, patients can receive functional tissue that mimic their native organs, thus reducing the need for organ transplantation. Another area where molecular biomaterials can be applied is in drug discovery and testing. Traditional drug discovery methods rely on animal models, which can often be inaccurate. By creating tissue models using molecular biomaterials, researchers can develop more accurate and predictive models for drug testing. This can accelerate the drug development process significantly and reduce the use of animal models. Furthermore, molecular biomaterials can also be used to study disease mechanisms and develop new treatment strategies. Through tissue engineering, researchers can create models of diseased tissues and study them in a controlled environment. This can provide a better understanding of disease mechanisms and lead to the development of more effective treatment strategies. Finally, the use of molecular biomaterials in tissue engineering can also contribute to the development of precision medicine. The use of personalized tissues can provide better treatment outcomes by tailoring the treatment to the patient's specific needs. This can help reduce side effects and increase treatment effectiveness. By combining molecular biomaterials and personalized medicine, TS can receive treatment that is specific to their needs and traits. In summary, advances in molecular biomaterials have enabled the development of more effective tissue engineering strategies. The continued development of molecular biomaterials holds promise for the repair and replacement of damaged or diseased tissues and organs, and the creation of personalized healthcare solutions. While challenges remain, the potential benefits of molecular biomaterials are significant and warrant continued research and development. Additionally, the use of molecular biomaterials has the potential to improve the efficiency and speed of tissue engineering. Traditional tissue engineering methods can take several weeks or months to generate functional tissues, making it challenging to meet the high demand for tissues. However, molecular biomaterials can accelerate tissue engineering by providing more precise and tailored substrates.